Ladies, gentlemen, folks, thank you for your attention. I'm Don Lindberg. I'm director of the National Library of Medicine, as you might have guessed. And this is always a lovely event for us. This lecture is sponsored both by the Medical Library Association and by NLM and the Friends of NLM, the latter offering you reception after we complete our work. Our speaker is a notable scientist and also, I think, has taken time out of a busy schedule to be with us, so we're grateful to you for that. He's at the Salk Institute, the Crick professor, and between him and Salk and Crick, dripping with scientific glory. How wonderful. We're lucky. Amongst his other achievements is I guess, crafting the concept, anyway, of this BRAIN initiative. At least so the White House is pleased to give you credit. So therefore, we're pleased to listen. And I'm hoping I won't delay your talk. I apologize to the audience a bit because we're starting exactly on time, just as if we were really all here and knew what we're doing. But in this case, it's because of this being video cast back, of course, to MLA and to the rest of the world. So we'll try to act professional henceforth. I must say, of all the things that you've done, this BRAIN Institute, while just beginning, is certainly a very, very interesting top note. And I wish you would tell us a little bit about what sort of computer and database support would be appropriate and what, if any, role NLM should play in it. Is that enough prodding? Let me welcome you to the podium. We're very glad that you're here.
Well, it was a great honor to be asked to give the Joseph Leiter lecture this year. And it's particularly appropriate that this is really on June 5th of this year, a committee that I serve on delivered to the NIH and was released to the public the final report on the BRAIN Initiative 2025. And this lecture is going to be, it has several different strands running through it. One of them is the BRAIN Initiative. Obviously, that's what's gotten the spotlight. But also, it's a personal journey, and it tells you a little bit about how we got there and my personal perspective on some of the, both the origins, the hope, and also looking into the future. What can we expect over the next decade? Well, if you look into the brain, you will discover that there is an alien technology that has gone into building the most complex device in the known universe. So your brain has more computational power than all of the internet and computers put together. But what makes it really remarkable is not so much how powerful it is, but the fact that it works on roughly 20 watts of power. That means it's built from a technology that is incredibly efficient. Supercomputers that cost hundreds of millions of dollars take megawatts of power. But somehow our nature has given us this remarkable device that is working at the molecular level, nanoscale technology, long before humans were aware of that. Now, if you look at the different levels of organization, starting at the bottom here at the molecular level, we know an enormous amount about the brain. We have a parts list. We know all of the different, for example, the different types of receptors at synapses. We know a lot about the ion channels and neurons. We also know a lot about the top here at the spatial scale here at the level of the system, the visual system. We have a good idea of how the neurons in the visual system respond to the world and how the long-range connections between them. And here on the right, you're seeing a new technique called diffusion tensor imaging that's giving us a sense for the long-distance communication between different cortical areas. So we know a lot about here with brain imaging, a lot down here at the molecular level, the part that we know the least about is the small, very densely connected network of neurons, the neural circuit. And that's really going to be the focus for the Brain Initiative, to try to untangle that very complex, very highly evolved and very, very sophisticated circuit in the cortex. There's 100,000 neurons in the cubic millimeter of the cortex, and connected with 1 billion synapses. So this is an incredibly dense, very, very intricate, and you'll see in a minute how we're going to be dissecting it, taking it apart, and trying to understand how it works. So if we take a step back and ask, where does our knowledge of the brain come from? It turns out that it's neuroanatomy, finding new ways, new techniques of being able to isolate and understand the individual neurons that make up the brain. And the technique that you're seeing here is called the Golgi technique, and it goes back over 100 years, back to the time of Ramon y Cajal. And this is a technique, reduced silver technique, that stains individual neurons almost in their entirety. And you can see here a cortical cell, the cell body, the dendritic shaft that contains thousands and thousands of synapses that are receiving information from other neurons, and then a very thin little axon that is coming off the bottom here that is sending signals, all or none spikes, to thousands of other neurons. So there's a tremendous amount of convergence from thousands of neurons, some computation that's going on in the dendrites, and then information comes out. Now this is an interesting picture historically, because this is not a photograph. This is actually a drawing that Cajal made, because he did everything by hand, right? He didn't have computers back then. The other thing that is misleading about this, although it's given us beautiful pictures, is that if all the neurons in this little piece of the cortex were stained, it would be completely black, because the neurons are packed together cheek to jowl. And the Golgi technique miraculously only stains 1 in 1,000. Otherwise, there'd be interference with all the other neurons. But we actually want to see all the neurons. We want to develop new techniques that will allow us to peer in and see how the whole thing is put together. And new techniques have been developed. I'm going to show you some results from my own research. This is a collaboration with Kristen Harris at UT Austin. And I'm wondering here if we are having a problem with... Here it is. So this is one apical dendrite from a hippocampal pyramidal cell. The hippocampus is a part of the brain that has to do with memory. And there are 75 synapses, each of them on one of these little spines that are coming off the shaft of the dendrite. And the little red area here is the active zone, which contains receptors. Here are the 75 axons that form synaptic contacts on those 75 little spines. And they're coming from 75 other neurons from other parts of the hippocampus. Now, it turns out that in addition to the neurons, there's something called astroglial cells. And this is just a small part of a single astrocyte. It's all interconnected, a single part of a single cell, and it is interspersed between all of the axons and dendrites within that little chunk of neuropil, which is about 5 microns across, 5 by 5 by 5. So this is still only a small fraction of all the neurons in this little patch, but we want to first explore the inside of that big dendrite. We're going to dip down inside of it, and we're reconstructing here the mitochondria, the copper-colored snake that runs through the entire part of this cross-section, and the endoplasmic reticula, which are shown here in red, are important for regulating the internal milieu of the neuron. And finally, we're going to come to a little tunnel. This is about 1/10 of a micron in diameter. It's called the spine neck. It was the thing that the spine head sits on. It's about 1 micron long. It's like a tunnel. And at the very end of the tunnel, you can see this region here. the postsynaptic density that contains the receptors that the neuron uses for communicating. So we're going to pop out and look back. And we are looking back. We were in that little tunnel, and that was the synapse that we saw. Here's another synapse. There's the axon. There's the receptors, and there's the spine. So in this little volume of tissue, there's about 400 synapses. So that's a tiny fraction of the 10 to the 15th synapses, but it really gives us a very high-resolution view of how they're related to each other. For example, here's the glial cell sitting right next to the synapse, and the glial cell is incredibly important because the transmitter at the synapse is glutamate, and the only way you get rid of it is by transporters on the glial cells. So without the glial cells, you're in trouble. Now, this is a little spicule here, and nobody has any idea what it's for. It looks like a little antenna, but they're fairly common. There are a lot of things that we still don't understand about this little piece of tissue. Okay, now what we're going to do is to reassemble the whole piece to sort of put it all back together. And I think we can bring up the music now. So here comes the rest of the dendrites, 150 of them, in yellow. This was all reconstructed from serial section electron microscopy, 100 sections from Kristen Harris's lab, and then it was all assembled in the computer. Here comes the 400 axons and axon fragments. And when it's all put back together, you can see it looks like a Chinese puzzle. It's all interconnected. It's an incredible piece of machinery. And one of the things that we could do is look at it, get rid of the dendrites, and we can see, like Swiss cheese, the little passages where the dendrite used to be. We can look at the extracellular space. We can begin to understand how things, molecules within the extracellular space can communicate between neurons. And finally, back to the astrocyte, the unsung hero of the cortical structure because of the fact that it plays so many different important roles, and the relationship between those postsynaptic densities in red and the astrocytes. These are partners. And finally, as the sound fades, we see here, we can take away everything but the dendrites. You can see those dendrites. And back to where we started with our central apical dendrite. And the intricacy of what's inside the cell is as complex as what's outside the cell, which makes it even more challenging. So what you just witnessed is something that has never before been seen by any human being until we reconstructed this. And it's now being done in many labs around the world. But before that, we had these pictures of neurons, which are very beautiful, but until we could reconstruct them completely, we didn't understand how it was all put together, and that's now an area called connectomics. And it's being automated so that we can do that for much larger pieces of tissue. Now, the story that I'm about to tell you goes back to the 1990s, the decade of the brain. How many people remember the decade of the brain? Well, you know, that decade came and went. And here's the problem. The problem was that there wasn't any central focus. It was clear the brain was important, but which part of the brain? Which disease of all the mental disorders and psychiatric and neurological problems that afflict humans, which is the most important? Who's going to set the priorities? And unfortunately, neuroscience, or maybe fortunately, is a very diverse community consisting of people at every level of that diagram, from the molecular at the level of single molecules, understanding how the synapse works, all the way up to the entire central nervous system, where we have behaviors being generated. And there's knowledge and problems and interesting research that needs to be done at all those levels. But if you have a national program, you have to have a focus. It has to have something that is like going to the moon. And the problem was that there was no consensus. And so therefore there was no moon. There wasn't any real program that could be made into an initiative, a national initiative. Well, that changed in a rather dramatic way just about 2 years ago, when a small foundation called the Kavli Foundation, which was founded by a Norwegian entrepreneur who came to the United States and created a sensor company that supplied the aerospace industry, gave his money to a foundation which produces, which has funded centers in 3 areas of science and engineering: neuroscience, nanoscience, and astrophysics. And there are Kavli prizes in these 3 areas that are given annually. But this small little foundation realized that there really ought to be a focus, and the focus that they decided would be one that could generate a consensus within the neuroscience community, was to develop new tools and techniques that would allow us to be able to speed up research. And by new tools and techniques, using molecular genetics, using optogenetics, all these new tools that are being developed, to improve them, scale them up, and so that way we could be able to use them to work on very difficult problems like Alzheimer's disease and bipolar disease, and be able to make progress in 10 years that would have otherwise taken 100 years. And we've seen examples of this happening, for example, in genomics, whereas the first human genome took 15 years and $3 billion, it now has reached the point where you can sequence a genome for just a few $1,000s, a factor of 1 million. And that has transformed research. It hasn't cured diseases, but what it has done is to set the stage so that we now understand the complexity of the problem. You've got to look at all the genes and how those interact with each other. So the goal is to do something like that for neuroscience. And the small group that I was a member of created this concept of a brain activity map, and the idea was to record from enough neurons in the brain simultaneously so you could begin to see how they cooperate with each other. If you record from one neuron at a time, which is the traditional way of doing it, and if you realize that there are about 100 billion neurons in the human brain, right, you can calculate how long it's going to take you. So if we can create new tools and techniques to record from more neurons at the same time, we'll be far ahead. And that was the concept that was embraced by the White House and announced on April 2nd last year. And the BRAIN is an acronym for Brain Research Through Advancing Innovative Neurotechnologies, and the important letters here are I and N, Innovative Neurotechnologies. That's really what the focus is. So I was in the White House, and this photo was taken just before the announcement was made, and it brings together the key players, that agencies and foundations that are partners and will be contributing. So first of all, I just want to point out that from the Kavli Foundation, the president, Bob Kahn, and the chief scientific officer, Myung Chun, they're the ones who brought the concept to the Office of Science and Technology Policy. And Tom Khalil, who was the person who really took charge, was very, very effective in bringing this to the notice of the president. But the lead agency is NIH, and Francis Collins has been extremely effective as a leader in pushing through what I think is an extraordinarily good approach to the difficult problems that we have ahead of us. I'll be coming back to that. But I want to also point out that, in addition, Cora Merritt here represents the National Science Foundation, another of the agencies that was announced to be a partner of the BRAIN Initiative, and Arti Prabhakar from the Defense Advanced Research Projects Administration, or DARPA. Now, in addition to these government agencies, and also I should mention that the President's Commission on Bioethics, which is also going to be very, very important in making sure that any of the research is being done is done at the very highest level of ethics. Amy Gutman is the chair of that commission. From UPenn, And then there are private partners, and that includes the Howard Hughes Medical Institute. This is Jerry Rubin, who is the director of Julia Farm. They're creating a brain initiative for the fly. And you may ask, why the fly? Well, it turns out that the fly has a small brain, but it has very sophisticated behaviors, and so understanding the fly is a nice first step. And then there is the Allen Institute for Brain Science. It's funded by the Microsoft founder, Paul Allen, and Alan Jones is the director. They're creating the brain initiative for the mouse brain, and they already have developed maps of the anatomical mouse brain. They want to do the same thing now for the electrical map, the brain activity map. And then finally, the Salk Institute. I was representing the Salk Institute that will be focusing on non-human primate research. The brain of the macaque monkey and other non-human primates are incredibly important as a bridge between these animal models, like the mouse and the fly, and the human brain, which ultimately is what we want to really understand. So what is the goal? Here's the overarching goal: to map the circuits of the brain, to measure fluctuating patterns of electrical and chemical activity flowing within these circuits, and to understand how their interplay creates our unique cognitive and behavioral capabilities. And so here's the bringing together of all of those parts. What's the real goal? The real goal is to understand how an individual brain works. Right now, we study the anatomy in one brain, and we study the recordings in another brain, and then we study behavior in another brain. It's very rare that you can do all of those at the same time in the same brain. But ultimately, every brain is different, so we've got to do all three at the same time. You've got to be able to record from enough neurons and know what neurons you're recording from. You've got to know how they're connected with each other, right? That's connectomics. And then you have to relate those activity patterns to behavior. And you have to be able to test the behavior causally, and the way that you do that is by perturbing the activity. That is to say, manipulating, either turning off or turning on activity in different neurons. And what makes it challenging is that we don't even know how many different types of neurons there are in the brain. We know in the retina there is on the order of 60 to 100. We think that in the cerebral cortex there may be up to 1,000. And we need to have genetic access so we can go in and manipulate each one of those cell types. And that's going to be one of the goals for the Brain Initiative, is to be able to put together a catalog of all the cell types of the brain, to be able to record from enough of those cell types to be able to understand how they participate in the function of the brain, and then to take that, map it onto the connectivity, and see how that generates the behavior. That, in a nutshell, is what we're trying to do. I was asked to be part of this working group, which reports to the advisory committee, to the director of NIH, Francis Collins. The chairs of the committee were Corey Bargman and Bill Newsom, and we represent all the different areas of neuroscience that are going to be important for this overarching goal. And I have to say that this was a humbling experience for me personally, because of the fact that there were so many issues that we had to struggle with, and we weren't in agreement all the time. In fact, I learned an enormous amount over the last 15 months, both about areas of neuroscience that I had previously had only very, very modest appreciation because it was outside my area. But I also learned a lot about my colleagues because they had their own views. And so we were able to understand and eventually come up with a consensus document that was released just about a week ago. I have to give special thanks to Corey and Bill for doing a magnificent job of organizing and ultimately crafting, because they had to put all of our words and contributions into a document that was readable, that was efficient and effective, and to really organize every aspect of the committee. Now, there were some guidelines that came from Francis Collins, and I just want to go over them very briefly, because they turn out to be very important and very wise in terms of helping us to organize our recommendations. Appropriate model systems, interdisciplinary research. It's absolutely essential that we bring engineers and physicists and mathematicians in to work with our biologists and neuroscientists, because they're the ones who have the ability to create tools, the ability to expand and scale up, and the ability to analyze big and very, very large data sets that Library of Medicine here has curated. And so without the partnerships that we have with them, without the teams, we're not going to be able to take on something as complex as the brain. The spatial and temporal scales are immense. I've showed you the spatial scales, that's 11 orders of magnitude. Well, the temporal scales go from milliseconds up to your lifetime, which is also about 11 orders of magnitude. So we're talking here about a huge space-time arena that we have to play in. Now, this is the part that I think is going to be critical, and I think it's where, if we don't execute this, the Brain Initiative is going to fail. And it has to do with accumulating larger and larger data sets, but putting them into a format and making them accessible in a way that will help the whole community. So the tradition right now in neuroscience is you get a grant from NIH, you get an R01 to do an experiment. You do your experiment. It may take, if you're working on a monkey, it might take 2 or 3 years. You analyze your data. You write your paper. You get back your rejection notice, and then you hopefully get it published eventually. And then, 5 years has passed, and then what happens to the data? Well, it gets put together in some archive in your lab, and never again sees the light of day. In fact, the graduate student goes away, and the PI can't even locate it, or even if the PI could locate it, can't figure out what the format was. And even if they could figure out the format, have no idea exactly how it was analyzed, because all of that was done, by the graduate students. The graduate students had the graduate students gone on to be a postdoc, and maybe the graduate student doesn't know anymore. So this is a terrible waste. It's kind of you've put all this effort into the data. Why don't you have it? And the problem is that it's considered personal property. The lab, it's my data. Sharing is difficult because you have to explain to the other person exactly the details of how you did the experiment and so forth. Well, here's what we got to do. We got to change that culture. And that may be more difficult than understanding how the brain works. We need to be able to create a platform where it's easy to download your data, where it's not a struggle, where you can share it with others. And ultimately, in terms of the Brain Initiative, we need to create these very, very large data sets that are so large that no single lab can analyze them, or no single lab actually is able to do all the work that needs to be done. In the same way that this has already been done for genomics, we have genomic databases, we have databases for X-ray crystallography, for protein structure. The physicists have created these data sets that are publicly accessible. Astronomers, Hubble Space Telescope. It can be done. It's going to be particularly difficult for the brain because it's so heterogeneous. It's not like ATGC, and it's not like stars that are fairly well understood in terms of their types and so forth. Although there are complexities there too, but the point though is that every single type of data that neuroscientists collect, ranging from activity, electrical activity, chemical activity, connectomic data, how things are connected, genetic data, behavioral data, all of those are very different in terms of the demands they make, in terms of how you are going to curate them and how you're going to make them available. But these are problems I think that we need to solve if we're going to really make progress and scale up neuroscience so that we can understand enough about the functioning of the brain so that we can then help figure out what goes wrong in the brain when we have schizophrenia or autism or other mental disorders. So just to give you a sense for what we did, we tried to be as thorough as possible because we really wanted to get the input from the entire community. And so we were willing to listen to anybody. In fact, people would, at meetings, right, would button collar me and say, "Terry, you're doing it wrong. Here's what you should do." And I was quite happy to listen to them. And in fact, it turns out that a lot of the ideas they had were really good ones and eventually got incorporated. But the point, though, is that without that process, then you miss things and you don't really have the right priorities. And so we interviewed 48 outside experts. We also brought in commentary from the public, special interest groups, for patient advocacy groups. We had a series of 4 meetings on topics ranging from molecular approaches to recordings, large-scale recordings, theory, computation, big data, which is an area that I have a particular interest in. And then the one that I think was the most interesting and for me the most revealing was human neuroscience. So we brought in clinicians. We brought in people who were actually in the operating room recording from humans with epilepsy in order to be able to isolate the focus. And there are humans walking around with hundreds of electrodes in their head for a couple of weeks waiting for a seizure. And so this is a fantastic opportunity if we could organize a team of people to actually go in and help the surgeon who has his hands full to help them figure out exactly the placement of the electrodes, exactly how to record and analyze the data, how to actually interact with the patient. And interestingly enough, the patients who were coming in for really intractable epilepsy that wasn't responding to any drug at all, these were patients who were, might think that, they were really under stress and they're really concerned. Well, it turns out that the patients are not only very interested in what we're doing, but they're very happy to help us because it gives meaning to the malady they have. It says that something good will come out of it. And so I think this is a fantastic opportunity that we have that we're not taking advantage of. Only a few labs around, hospitals around the country are prepared to do this, and it's a very difficult, complex, expensive task. If there's nothing else we can do with the Brain Initiative, it's to be able to help those patients and those teams that are trying to understand what's happening inside the brains of those humans. It really is something that is, I think, going to be even more important than a lot of the animal models. I think the human model is, the human is the best model system for the human being. Okay, well, the final report, 146 pages. It's not light reading. It's not the sort of thing that you're going to sit down with on a weekend and plot through it. But I can tell you that I'm getting reports back that colleagues of ours in China and in Europe are looking at it line by line because they want to create their own brain initiatives in China and Japan. And they're using this as a template for understanding, what the priorities are. I'm not going to go through these except to say that we did our best to come up with the most promising areas where we think if we could put some resources, we could really make an important avenue for technologies to improve the ability to be able to both record, figure out the connectivity, understand fundamental principles based on the analysis of the data and making models of the data, and ultimately putting them all together, integrating together all these technologies. And again, that means bringing together talent from many different areas, many different disciplines, from many different labs, changing the culture of neuroscience as well as the techniques and technologies. So let me give you a little taste of what we're talking about. This is a really interesting graph. It's data, but it's not the kind of data that normally you'd find in a scientific journal, right? Normally, you do an experiment, and a little data point was the value of cyclic AMP as a function of time. Well, this is time. Each dot here turns out to be a journal publication. So this is a paper, and the y-axis here on a log scale is the number of neurons that were simultaneously recorded and reported in that paper. Now, this all starts back in the 1960s when Hubel and Wiesel and others at the time were mastered the technique of recording from one neuron at a time using a tungsten microelectrode. A fantastic advance, and it really set the stage. And really, in terms of the visual system, it's the microelectrode launched. Having that technology just launched hundreds of labs that were completing the job of recording from all the different parts of the brain. And as you can see, as time went on, it was possible to record from more and more neurons. And indeed, on a log scale, this is going up exponentially, so that by 2010, we were up to roughly 100 neurons. And so it's possible to record now, like in the hippocampus of the rat, from 100 neurons as the rat is running around in an arena. Now, there's still a drop in the bucket, right, because there's 1 million neurons in the hippocampus of the rat, but, at least it's more than one at a time. But the real problem here is the slope. And this is on log scale, it's the doubling time. It's telling us that we have to wait 7.5 years in order to increase the number of neurons recorded simultaneously by a factor of 2. And that's really taken us, 40, 50 years here to go from 1 to 100. Well, how many neurons do we want to record from? I submit that 1 million is a really good number. Why? 100,000 neurons in a cortical column. You want to record from, say, 10 of them, different parts of the brain. That's 1 million neurons. Okay, how long is it going to take us at this rate? So I've extended the graph out here now. Let's extrapolate from where we are today, here, out to 1 million, here, 2,100. Boy, in 2,100, I will guarantee everybody in this room will be post-Alzheimer's. We can't wait that long. We need to speed things up. Okay, so how do we do that? Well, with new technology, the hope is that we can speed it up by a factor of 100, 10 to 100, and we can get to that by the end of, say, 2025 to 2030, right? Now, that's really asking a lot, because it really means that we have to come up with new technologies. Now, there's a ray of hope, because it turns out that, meanwhile, technology has been improving. So I've already mentioned optical recording. That's a new technology that's just surfaced in the last 10 years and has revolutionized our ability to record from neurons. From genetically encoded calcium dyes, we can look at calcium activity. It's not the same as electrical activity, but it's a pretty good surrogate. It tells you the cell's firing at a high rate. And with that, now Mark Snitzer holds, I think, Mark Snitzer holds the record. He recently reported that he was able to record from 1,000 neurons. So we're already up here, right? We're 2014. We're right on track. So we just need 1,000 Mark Schnitzers now, and we'll get to 1 million. Now, sometimes, it's really interesting, sometimes things happen that are completely unexpected that suddenly change the game completely. And such an event occurred while we were writing our report, actually, which just blew the top off the whole goal that we had, right? So our goal is to record from enough neurons that you can understand what the behavior is, right? That's kind of kind of a simple encapsulation of the ultimate goal. Okay, let's pick an animal that has small enough neurons that you can actually record from maybe most of them, but has sufficiently complex behaviors so that you can actually begin to see how the behaviors can be modified by the environment, and you adapt and so forth. And it turns out we have such a creature. It's called the zebrafish. And the zebrafish has one big advantage going for it. It has a transparent brain. That means you can see right through it. Our brains, and all mice brains and mammalian brains, they're not transparent, and that means that you can't see very far in them. That, you look into them and they're opaque. And with optical techniques, you can get down maybe 100 or 200 microns, but the mouse brain is a centimeter across, and so you're stuck. But if you have a transparent brain, you can just look right through it. So I'm going to show you a movie, I hope, which is really extraordinary because it's giving us a taste of what we're going to be able to do within the next 10 years. So what you're seeing is a zebrafish brain, and each of these little dots is a calcium signal in one neuron. And you can see that they're flashing on and off. Now what's remarkable is that they were able to record, this is Misha Ahrens at Janelia, he was able to record from 80,000 out of 100,000 neurons, 80% of the neurons in the brain. And this is a brain of an animal that was immobilized, so it's not doing very much, but it's clearly doing something. The brain is doing something. What is it doing? And you can see these flashes of activity going down the spinal cord here on the right. Let me try that over again. And you can see that there are, these are coordinated. It's not like they're random. And you can see that there are times when there's a surge of activity, like there. What is the zebrafish thinking? These are internal states. It's like, you know, what happens in your brain when you're sitting there immobilized and thinking, right? Activity patterns, flashes. So here's the problem. It's a Chinese wish. May you get what you wish for. It's a Chinese curse. And so now we have the problem. How do we decode what we're seeing here? We have the tools, and now the challenge is to go in and see how to manipulate these patterns of activity and allow us to be able to ultimately relate them to behaviors, and then ultimately to see what happens when different parts of the brain go bad. For example, the hippocampus. We know that if you lesion it, that you have problems with long-term memory. Well, how exactly are the signals between the hippocampus and the cortex regulated? We'd like to be able to use these tools to be able to answer that question. Now, I've already mentioned that the real problem here is scaling up. How do you go from, you know, a 1 neuron at a time to 100 at a time to 1 million at a time? That's scaling up. And that turns out to be something that we're very fortunate to have a new tool that helps us do that. It's called machine learning. Let me explain to you what that is. Well, we record from 80,000 neurons in the zebrafish brain. And you can't use the old technique of plotting for each cell the tuning curve, because there's just too many cells. So you've got to come up with new ways to deal with it. Now you're dealing with a space that instead of having 1 or 2 or 3 dimensions, it has 80,000 dimensions. Just imagine. You can't even imagine how, you know, what that space looks like, because you know, we can only think in 3 dimensions, maybe time 4. And so we need new tools, mathematical tools, for exploring spaces of high dimensions. And that's what machine learning has accomplished. It has developed over the last 25 years and has developed mathematical and algorithmic tools that allow computers to search through these spaces for patterns of activity that the humans can't see because it's just too complex. Just the way that you couldn't see what was going on in that zebrafish brain. Using the right machine learning tools and techniques, you can actually tease out things there that you can't see. In the same way, scaling connectomics from that 5 microns, cubic microns, up to a millimeter cubed requires machine learning techniques to be able to do the segmentation in the EM and the reconstruction in 3D. That turns out to be very labor intensive. It requires that human beings sit there and do it because it's a very difficult pattern recognition task. And it turns out, again, that you could teach a machine learning algorithm to do that by shadowing the human and figuring out what the human is doing and to be able to replicate that and scale it up by a factor of 100 or 1,000. Behavior, and this is the part that is very, very complex because of the fact that, you know, behaviors have so many different dimensions in terms of how you approach a visual stimulus and how you react to it and all the different ways you can deal with it. And you know, the way that neuroscientists have traditionally dealt with it is by reducing the complexity of the task, sitting a monkey down in front of a screen and giving the monkey the option of 2 different choices. You know, either it's going to the right or the left, and the stimulus is going to be very simple. Maybe you vary 1 parameter, and you need to do that because of the fact that there's just too many things that can be influencing the brain, and you want to reduce the dimensionality to something you can analyze. Well, the problem is that if you want to understand the brain on its own merits and in terms of what it was actually designed to do, the brain was not designed to sit there and press a button. The brain was designed to interact with the world. It was designed to interact with other human beings. It was designed to be able to make decisions in a very uncertain environment where there are probabilistic outcomes. And how do we study that? Well, interestingly, it turns out machine learning can be used to analyze behavior. And it has been used, for example, to analyze the behavior of flies. So flies do a lot of different things. You know, they walk around, they fly, they interact with each other, they communicate, they do funny things with their wings, they groom. Well, it turns out that you can automate that. In the past, humans had to sit there frame by frame and figure out what the fly was doing. Now you can automate that by using machine learning to go through the frames and automatically indicate what's going on in each frame. In fact, using unsupervised techniques, it's possible for the machine learning system to discover new behaviors that flies have that were never seen before, because either they're too fast or they're too complex. But you can now mine the database and find out what's really happening. And that has been done for mouse behavior. It's been done for human behavior. So one of my former graduate students, for example, who was interested in human facial expressions, has used machine learning to automate the detection of the different muscles in the face, collaborating with Paul Ekman. And now we have a company called Emotions. Oop, is this a conflict of interest? Well, be that as it may. But the beauty is that it can be done completely automatically and objectively and done in real time. So this is going to really have a huge impact in many different areas of medicine where you're trying to find out what a patient's really feeling and thinking, and the words are disconnected from what's really going on. And so there's a lot of applications that are going to be very important when two people are communicating. What's going on in their face tells you a lot about what they're feeling at the same time that they're saying. So this gives just a few examples of the sorts of things that machine learning might be capable of doing, and just to give you a sense for how important this field has become. The premier meeting is called Neural Information Processing Systems. Interestingly, started by a small handful of neuroscientists who were trying to figure out how to deal with the problem with scaling up the recordings from neurons, but now has become a very major topic in computer science. And at the meeting, it was held last December, over 2,000 participants from academia, from business industry, all the major leading information companies, Google, Amazon, Facebook were all there. And they were there enforced because they were recruiting. They were trying to get students to come and help them mine their databases, right? They have huge databases of what people are saying and doing and thinking that they put online, Twitter, Facebook. And in fact, one of the workshops on deep learning was so popular that Mark Zuckerberg from Facebook showed up and announced that they were starting a new lab, AI lab, that one of my friends, Jan LeCun, was going to be leading, and they were going to be hiring. So that was an indication of the importance for them. And just to give you a sense, a feeling for what's happening with deep learning, and by the way, how much time do we have left? Okay, I think I'm on track. I just want to give you a sense for what's happening, because this has really had an enormous impact on areas like speech recognition, object recognition in images. I already told you about behavioral tracking. Basically, whenever you have large numbers of signals, very high dimensional spaces, these new tools and techniques that are becoming available are able to really dissect the data in a way that you can use it in a very, very direct and in some cases profitable way. So here's the architecture, and this actually goes back to the 80s when neural networks were developed. Neural networks are very simplified versions of brains, right? Where a single unit here corresponds to a neuron, but a very simplified neuron that could only have a limited range of outputs. Didn't have dendrites, but it did have connection strengths called weights. But the idea is in 1980, we could only do 1 or 2 layers here and only a few hundred neurons. And so it wasn't possible to really solve complex problems, although it looked like it was doing better than the traditional AI techniques that use logic and symbols. Fast forward to 30 years, and Jeff Hinton, who's a colleague of mine who I collaborated with, has been one of the leaders in this field in developing deep learning. It's now possible to train networks that have 12 layers deep, that have 1 million model neurons, and the reason why it's possible to get these networks to train up a billion connection strengths is because the databases have gotten enormous. In other words, you need lots and lots of examples. So the key to success is seeing lots and lots of cases of each individual thing you want to recognize. If you only have 1 or 2, how do you generalize from that? You have to generalize from just a few cases. But if you have 1,000 cases, it's easier. So what they did was they used YouTube input. And they trained up this large network, and as you can see when they started analyzing it, that in the earlier stages you get things like these what are called Gabor filters, which are found in area code in primary visual cortex area V1. And this has been well established to be important for biological vision. But in the latter areas, you start coming up with units that have much, much more specific properties. And in fact, in the case of the monkey brain, we've found that there are single neurons in the monkey brain which recognize faces, in the sense that if you give them particular faces, the neuron will fire at a high rate, but if you give them houses and coffee cups, they won't fire at all, right? And there are face areas where there's a concentration of these neurons. So we know that these things exist in the monkey brain, and lo and behold, in this data set from YouTube, there are a lot of faces. And indeed, you find units that look as if they are recognizing faces. Isn't that interesting? But it turns out that the most common unit that they found at this very highest level turned out to be cats. And it's telling us, okay, this network is telling, it's synthesizing the world's movies of all the things that are out there, you know, random movies, and it's synthesizing what is there that is in common. What is there that is a recurring theme that is going to be important for perhaps making discriminations, being able to recognize these objects. Once you get units like this, it's a lot easier to recognize a face by using a very simple regression output. Now, it's not just the NIH. DARPA is also engaged, and they have two big projects. One of them is to improve deep brain stimulation. The electrode that is being used right now in 100,000 subjects, these are Parkinson's patients, is a device that was approved 30 or 40 years ago. It's like a crocheting needle with a big tip. You know, it's a kind of like a cattle prod. But it's effective and in alleviating a lot of the symptoms. Well, we could do a lot better now. If we just took the existing simulation techniques that are used in monkeys and approved them by the FDA and used them in humans, we would vastly increase the capability to be able to help people who have problems, not just from Parkinson's, but from depression and from other types of diseases, mental disorders, for which activating neurons and pathways might help. They also have a very, very ambitious and almost science fiction goal here with memory prosthesis. DARPA has been very effective in helping with limb prosthesis. They've developed robot limbs that can be activated by motor cortex. So this is directly from the brain activating a robot limb. And that's been very, very important for helping soldiers coming back from the Middle East who have lost their limbs. But what about memory? They have traumatic brain injury. They're coming back with problems with memory, problems with concentration, attention. Could we, using the same tools, recording and then stimulating, help memory? And that's the goal here. And Jeff Ling is the director of the new Biological Technology Office that is organizing these projects. And I'm going to quickly skip over this, except to say that Helen Mayberg has been one of the leaders in figuring out which part of the prefrontal cortex, which when you stimulate it, can alleviate patients with clinical depression that are resistant to any antidepressants. And so that's research in progress. There are still clinical trials going on, but I think this is just the tip of the iceberg. I think that this is going to develop a whole new technology that will be able to very, very accurately and specifically activate particular types of neurons in different parts of the brain. NSF has its own, I should say not problems, but projects. There was a meeting that was organized in May, a little workshop that I helped organize. We had a list of 30 people that we invited, and 140 people showed up, to show you how popular and exciting this was. It was right after the announcement in April. So here's a program called EAGER, and the idea is to look for innovative ideas. And so NSF wants to help get new ideas that no one else has explored before, and see if we can search through and find new ways of solving these difficult problems. Interestingly, the state of California got wind of the BRAIN Initiative and decided, California, we have the 6th largest economy in the world, maybe we should have our own BRAIN Initiative. And so last year, Ellen Corbett, who's the head of the committee that looks into this, visited, had a hearing, and right now they're putting together the state budget, which may include a California BRAIN Initiative. And finally, and this is something that took a long time for me to understand, and this is something that biggest question I'm always asked is, what is your big, project that you can sell to the American people? Usually it's a disease. We want to solve Alzheimer's. That's something everybody understands. And the past projects, for example, sending a man to the moon, had that kind of iconic status. The idea of a man standing on the moon somehow resonates around the world. Everybody understands that. But we don't have something like that. And in fact, it would be a mistake to use a single disease as our goal, because billions of dollars are already being spent to try to alleviate Alzheimer's. And progress is very slow and painful. Okay, well, it took me a while to figure out that, in fact, the marquee that's used to fund these projects may not be the most important impact that they eventually have. Let's take a look at the man on the moon. Yes, it's true. In 1969, Neil Armstrong set foot on the moon, but have we been back since then? No. Why not? Well, there's actually not much there to go back for, right? A few moon rocks. But why was it important? It was important politically, because we were in a race with the Russians. Number two, it created an industry, a space industry. Satellites that now are circling the globe, that are used for weather prediction, for GPS, and for lots of other things, right? And it's a big economic part of, it's a big chunk of our economy. So the moon itself was not the real goal. The real goal was to create an infrastructure that could boost the engineering capabilities, and not just in rocketry, but also in electronics and materials. War on cancer, right? That was a big one, and in fact, you have a whole cancer institute here. Well, did we cure cancer? That was our goal? No. Okay, but what was created was recombinant DNA technology, which again allowed us to manipulate genes and created a whole new biotech industry. Again, it was the economic payoff that really was the ultimate impact. It wasn't the solving the problem that you thought you were solving. It was the journey, what you needed to solve the problem, the tools you needed to solve the problem. That was the payoff. The Human Genome Project. I remember that the goal was to cure human diseases. Remember, this was how it was sold. But the fact is, once you have the whole genome, you can read it all you want, but it's not going to tell you how to solve the disease. That's going to require a lot of research. It's going to help you. And in fact, it has created a whole genomics industry. In a study that was done by Battelle, they discovered over the years that for every dollar that the federal government invested in the Human Genome Project, that there was $140 of economic activity that was generated because there's new companies. A small little company in San Diego that was started about 15 years ago called Illumina is now a $100 billion market cap. They have the best gene sequencers. And it's changing medicine, individualized medicine. It's changing the way that we biologists think about the problem. It has a huge impact, but it's not the ones that were used to motivate why this is such an important project. What about the Human Brain Project? What's going to be the payoff? Well, we want to understand how the brain works, but that's a really difficult problem. But I think that the real impact is going to be the creation of a new industry as important as the space industry, as the biotech industry, and as the genomic industry. It's going to be called Neurotech. And it's going to be device industry, just as pharma has created a very, very sophisticated drugs that can be used to alleviate lots of illnesses and very effectively. Those haven't been as effective for the brain. Why? Because unlike the liver that only has a few types of cells, the brain has thousands of cells. And there are all these side effects. But if we can go in with electrical stimulation, optical techniques for being able to turn neurons on and off, we might be able to then use electricity and light to cure diseases in a way that is more specific and targeted and more effective. But to be able to do that, we have to create a whole industry, because that's dealing with the level of complexity that pills just can't approach. This is a prediction, and we'll see how that pans out. And finally, I think there's another payoff here that I think may be even more important for our country. This was an announcement that was made by John F. Kennedy in 1961 at a joint session of the Congress, and it was very audacious. We are going to the moon. And this was a time when the Sputnik was overhead, and the American public was really feeling kind of, what happened? I mean, I thought we were the head of, the head in terms of engineering and science. But But Kennedy was very bold and said that, we're going to go within the next decade and we're going to do this. And he inspired the nation. And when Neil Armstrong got to the moon in 1969, the average age of an engineer at NASA was 26. Let's think about that. That means that 8 years previous to that, they were in school. These were young kids that were just deciding what they're going to do with their lives. They were inspired. They wanted to be part of this mission. They thought that they could, this is a career path for them. And how exciting and important that was, it really was an enormous undertaking that we just actually don't even appreciate today. The fact that, I'm not sure if we can afford to go back to the moon. But they did it. They did it. And they did it. And in the process of doing that, they inspired a nation. And that's what leadership is all about. I'm going to end on a scene that took place recently in a classroom. This is a colleague of mine, Tony Zador, at Cold Spring Harbor Labs. And as you can see, he is teaching a bunch of young recruits. I don't know if you can read it, but this little tag says Dr. Michael, and this is Dr. Victoria. Right? He's telling them about how the brain works. And in fact, he's actually illustrated it on his own head. The occipital lobe and the parietal lobe and the frontal lobe, right? This is the future, right? Who's going to put the man in the mind? It's going to be the next generation. In other words, we need to inspire a generation of students to go, the best and the brightest, to go into neuroscience, to go into trying to understand what is happening in our brains when we think about problems, when we make decisions, how to understand what happens when those decision-making processes go wrong. For example, when you're addicted to a drug, we should be able to figure this out. We should be able to go in and help these people. And we're only going to be able to do that if we can marshal the best that we have here in the United States in terms of human resources, in terms of engineering talent. And it has to be done in a way that is integrated into our existing scientific program. And I think that's what NIH has done with the BRAIN initiative. I think our report provides a guideline for how to do that. Now, it's all about execution, and the future is going to depend on leadership. And Francis, I think, has been a superb leader. There's a story behind this. Just before this photograph was taken, you saw the photo op that was taken a little bit later. I was talking with Artie, who's just out of the photo here, Prabhakar from DARPA, and we were exchanging information about the committees we were on, and she said, well, Jeff Ling is heading up our group in Brain Initiative, and I said, well, he should be ad hoc on our committee, and so we that's how you network in the White House, right? But in any case, as I was talking to her, suddenly she stopped talking, and there was a silence, and and suddenly I felt a tap on my shoulder, and I turned around, and there was President Obama, and the only thing I could think of saying, I didn't introduce myself, the only thing I could think of saying was, thank you, Mr. President, and thank you.
This has been a wonderful talk. It really was inspiring, as you intended it to be. I wish that others could have been present. Leiter himself was an interesting guy. He started out in cancer research and ended up being director of library operations here for many, many years. He would have loved to have been here. Thank you so much. This will memorialize your visit and also bring thanks of the library and the Medical Library Association.
Well, thank you very much. I appreciate the opportunity and the honor to be here.
Thank you. All right. In Washington, it doesn't count unless it's on TV. Thank you. Now, you had agreed you might take a question or two?
Oh, I'd be happy to answer any question.
Good opportunity, folks.
Or if you need to go...
I have a question.
yes, go ahead.
Dr. Sejnowski, You mentioned that one of the goals of the BRAIN initiative was to establish platforms for sharing data. And you know that we, as medical librarians, understand the daunting amount of data and images that are going to come from this project. And we're wondering if you can expand on that goal and how we can help.
Happy to. In fact, I'm the one who wrote that part of the report. So if you have any problem with it, contact me. So here's what needs to be done. And I think that it's something that Barry Richmond, for example, knows about, which is that he was about to escape, but that's okay. He knows that that the only way that you're going to get a neuroscientist to give up their data is by incentives. And so here's the incentive. Okay, I already told you that we have trouble backing up our own data, just backing it up so that we could look at it 5-10 years up the road. So let's do the following. National Library of Medicine, you have experience with making big data platforms. Help us create a platform so we could back up our data. And that means making it as simple as a few clicks on the internet, and boom, the data goes up. It has got to be in the right format. It's got to have the right metadata to go with it so you can interpret it. But the point, though, is that it's your data, and it's in the cloud, and you're curating it in terms of knowing what's there and so forth. But you can control the access. So you can control who, maybe just people in your lab or somebody you're collaborating with, but it's limited in terms of who gets to see it. But the point, though, now is that it is in a format that can be compared with other data sets. And at some point in the future, when the BRAIN initiative ramps up, and in our proposal, in our report, we predict, again, it's a big extrapolation, that this would ramp up to, say, 300 to 500 million a year over the next 5 years. But when it gets to the point where you really have these mega data sets, or giga data sets, or peta data sets, that no one group can actually handle, then what you've got to do is make it a restriction that when the data, when the paper is published, that it becomes accessible to everybody in the world. And that's what's happened with these other data sets, genomics data sets and protein structure data sets. And that really changed the whole culture. Once you do that, then you can begin to compare data sets. And here's a little anecdote I heard about from the Hubble Space Telescope. Half the papers that are now being published from data from the Hubble Space Telescope are not coming from the labs of the PIs who actually had the experiments that collected the data. It's coming from archival data that has been put into the public domain. And how is that happening? Well, it turns out that they had to put a lot of effort into calibrating it. So now it can be compared with new data that's being generated, and you go back in time and you see how the star was it a, if it was a supernova, what was it like 20 years ago? If you didn't calibrate it, you wouldn't know. And we got to do the same thing with brain data. But the first step is to create the platform, and that's where we need your help to be able to figure out how to do that. It's going to require a consensus, a format, a set of rules. How do you use it? What's the access? And if somebody does use it, what are who gets to publish and so forth? Very complex problems that have to be solved.
Thank you.
Hi, I enjoyed your talk very much. I'm a computer scientist here at NIH, and I have a great deal of respect from the early pioneering work you did with neural networks and people like Paul Verbosch and Gail Carpenter and Stephen Grossberg. I'm having a lot of trouble understanding these deep learning neural networks with all these layers and all these neurons and so forth. It seems to me like you might get spurious findings out of them, but it looks like they're over-parameterized and that sort of thing. Can you help me understand that better?
Well, that takes me back 30 years to the neural network days. And you're absolutely right. Any good statistician would tell you that you're crazy to have a model with 1 billion parameters, because there's just no way you're going to constrain it, right? You're going to overfit in the jargon. But that means that you're going to memorize. And how do you counteract that? Well, that's why you need more training data. The more training data you have, the less overfitting. But even there, it's still a miracle. It's still a miracle. We don't understand mathematically why they're so as effective as they are. And when I say effective, they're literally 30% better on all these different tasks in speech and imaging and object recognition than the best engineered system. So it's really, there's the secret of neural networks that we didn't know back then was that they scale. The bigger they are, the more data they have, the better they do. And there's no other tool or technique that's ever been used that didn't poop out after a while and didn't scale up to the larger and larger difficult problems. But here's why I'm fairly confident that even though we don't understand it mathematically, that we will someday, and why I think we in fact are on the right track, is that nature was there before us. Because if you look into the brain, you find these deep hierarchies. It turns out in the visual system, there are about 24 different visual areas in the cortex, and they're stacked all the way up. And it's in fact at the highest levels that you see these very, very specific neurons that respond to faces and objects. And my favorite one is a recording that was made at UCLA by Ishtak Fried and Christoph Koch. And they were recording from one of these epilepsy patients. And they were recording from a part of the brain near the hippocampus. So it's near the area that is important for long-term memory. They recorded from a neuron that would only respond to pictures of Jennifer Aniston. And not any other actress. Although I think it did respond to a picture with Brad Pitt at the time they were going together. But it's an anecdotal finding in the sense that they didn't look at, they had a limited number of images, maybe a few hundred that they could use in the limited time they had. But the point, though, is that in the brain, which has 10 to the 15 parameters, nature has somehow found a way to be able to create learning algorithms, real ones, and not just one, but dozens, which working together allow us to do these amazing feats of being able to learn things, languages, thousands of faces, many different facts. I mean, in fact, it scares me to think, what's the probability that that's the only Jennifer Aniston neuron in that brain, right? There's probably thousands, tens of thousands. Just what do you think is in your brain? How many Jennifer Aniston neurons are there? Or Brad Pitt neurons, right? It's a little scary, but in fact, that's what you need in order to be able to survive in the world, to be able to deal with the complexity of the world. So the answer is the jury is still out in terms of where that technology is going, but all I can say is that there's a huge investment that's being made by the information industry to exploit that. And we'll find out very, very soon, because as soon as, and by the way, Google has more computing power than any other company in the world, right? So they're the ones that are going to make these discoveries. And Geoffrey Hinton, my colleague, and I mentioned him in the talk, recently his company was bought by Google, right? And I asked Jeff, I said, well, you just sold out. Is that the end of your academic career? And he said, oh no, Terry, you don't understand. If I were at Toronto, at most I would have a computer that was kind of a middling road GPU farm, Here at Google, it's like, the sky's the limit, in terms of the research. It's going to go 1,000 times faster here. And the only problem, he said, was that the graduate students weren't so good because there weren't any. Yes, Doug.
After October, all NIH-funded research shall use both equal numbers of males and females and compare and contrast the data. How will that affect the BRAIN initiative?
Oh, what an interesting question. Well, we need more female brains, it's clear. Both in terms of studying them as well as female brains working on the project. I understand that there are complexities having to do with hormonal cycles and so forth. It seems like we could solve those problems, right? Now, the brain is an incredible device. It responds to hormones in a big time. In fact, one of the amazing things that was discovered was that during the estrous cycle, that the number of literally the spines and the synapses in the hippocampus is also cycling, which means that there's massive reorganization that's occurring. And that really makes it a little bit difficult if you're trying to understand the hippocampus. But I think that's of interest in itself. How does that happen? Maybe that'll give us a clue to how synapses are formed and how is it that you can maintain your memory over that cycle. So I think that there's a great opportunity here, not just a problem that we have to solve. Well, I'm impressed that he was able to learn how to shake with his right hand. Handedness is something that has a very long literature, and it seems to be related to hemispheric specialization. So we know that language tends to be, not everybody, but in the left hemisphere, and that goes along with being right-handed. But it turns out there are people who are left-handed, and of those, the language is not always in the left hemisphere. So it's a very complicated interaction. There's a question of how is it determined genetically or through experience which hand you're going to have. It turns out that there are some people who started out ambidextrous and because of convention are forced to write with their right hand. And so if that's done early enough, the brain circuits will reorganize, and that's an amazing thing. The ability of the brain to adapt and to create new circuits is something that helps us not only for normal function, but also after a stroke or some other brain injury, allows us to create a new circuits that allows us to get around the bottlenecks that are created by the stroke. So all these problems are interrelated in the sense that if we understood something about stroke, that might help us understand something about how the brain adapts to other traumatic experiences. And if we understood something about left-handedness and right-handedness, maybe that'll help us understand something about stroke. But without the tools and techniques to go in and see what happens in the animal brains under those different conditions, then we'll forever be sort of circling around and looking at it and saying, gee, there's something going on there we don't understand, but we don't have the tools to get at it. I think the next decade is going to be the golden period for neuroscience in terms of being able to answer a lot of these questions, because until now we didn't have the tools, but we will have them. In the back.
What do you think of the European Blue Brain Project's approach at reconstructing the brain from the bottom up with computational neurons?
Well, I'm not a physician, but I would say that if a brain was blue, that it was having problems. And you're referring to both not just the Blue Brain Project, which was the Henry Markram Project, but also the European Brain Project, Human Brain Project. And I think that that's a real contrast with the way that the Brain Initiative was organized here. Although the concept, comes from the president, he put it into the hands of the scientists to decide what would be best. And it came from the bottom up. I gave you the kind of the history of how that happened. Right? In the European Union, it was done exactly the opposite. Henry Markram had this grand concept of creating a model of the brain, and somehow he managed to sell that to the European Commission. And and it was a competition. There were other projects they could have funded. And by the way, the same thing is true here of the Brain Initiative. The history is very interesting. It turns out that the president had a menu of 12 things that he could have as a signature science and technology project. The brain was just one of them. And there are others like getting an asteroid around the moon, right? Each of his departments and cabinets had their own projects, a better battery. These are really good projects. I'm not sure about the asteroid project, but he picked the brain, and the European Union picked the brain, and I think that tells us something. However, when you do things top down, things can get awfully distorted, and it unfortunately never really went through this process of vetting and integrating. And as a consequence, they're having growth pains now, getting the thing off the ground. So I'm hoping that they'll be able to recover from some of these startup pains. But it's a very different project. It's complementary. And in fact, more than half of the funding is not going to go toward neuroscience data. It's going to go toward the computer science infrastructure for being able to integrate the data. So a lot of it is for high-performance computing. The Blue Brain Project, for example, was an IBM. It was called Blue Gene, the computer itself. That's why it was called the Blue Brain Project. And it's clear that they're going to put their money on computer science. We're going to put our money on sensor technology and big data analysis. And so, I think we'll have a way to bring on board all the different initiatives. In Japan, it hasn't been announced yet, but friends of mine who have been helping to organize the Japanese project tell me that they're going to go for a transgenic non-human primate, probably the marmoset. So the idea is to create that as a genetic model like the mouse has been for medicine. But we need to have a transgenic primate because the primate brain is a lot closer to our brains and has similar kinds of social interactions and so forth. So, you know, that's a huge, I mean, in terms of the staggering amount of work that has to be done to create a transgenic. It's gotten easier now because we have recombinases like CRISPR systems. So it will be possible to create knock-ins and knockouts in the primates, but you have to have colonies of hundreds and thousands. And that's what they, I guess, are able to do in Japan. One more question. Okay.
Hi, thank you. I just wanted to know, where do you think the ethical concerns could be in the future around knowing a lot more than we do now about the brain, especially in the realm of being able to recognize what a person is thinking or feeling by their facial expressions, and who has the right to go into your brain or my brain and then do this kind of complex analysis, and just wondered what your thoughts are on that.
Well, you're absolutely right that the technology that's being developed can be misused as well as used for good. And so there has to be a lot of thought put in ahead of time for unintended consequences. And this is something true with all technologies. It doesn't matter, what you're dealing with. Internet technology, right, has unintended consequences. So there's nothing bad about that. It's just that you have to really put some effort, and that's what the President's Commission on Bioethics is doing. They came out with a report just about a month ago in which I testified there, so you can go and take a look and see what I said. But I think, though, that there have to be real privacy concerns here. And that's already happening, by the way, with human brain data, is that you have to depersonalize it before you can make it available for other researchers. Not as easy with genetic data because there are ways of cracking that code. But we really hope that the research that's done and the consequences are going to that the immense benefits that could accrue will more than cancel I hope the misuses that are inevitable with any technology. Now, but I'm less concerned about brain reading because I think we're so far away from that it is still science fiction. Although it is true that there are some groups now that have done something which is pretty impressive, which is that by looking at brain imaging, which is very crude in terms of time and space resolution, but they're actually able to reconstruct what you're looking at by just looking at the activity in the visual cortex. That's kind of maybe very early stage of processing. I don't think you're going to be as easily able to interpret what's going on in your hippocampus, which is very abstract, and it probably has nothing to do with the actual sensory data. It has something to do with internal states, and that's really the challenge. The frontier is to understand how internal states in the brain are organized and how they make, through spontaneous activity, create new thoughts and ideas and creativity. That's the challenge, but we're not there yet. So thanks again. You're very welcome. Thank you so much.